Hello everyone, welcome to another video of biology concept. In this video, we will find out that which one is hereditary material, DNA or protein. We will see that how scientists discovered hereditary material. Let's start. In 1910, chromosomal theory of inheritance had been confirmed that chromosome is the vehicle of heredity for all the living organisms. But chromosomes contain both DNA and protein, so scientists were confused that DNA or protein, which one of them is responsible for inheritance. So they were doing research on it, and one of the scientists whose name was Frederick Griffith, who was a British microbiologist, in 1928 he made a series of unexpected observations when he was experimenting on Streptococcus pneumoniae. He found out Streptococcus pneumoniae in two types. One is S-type and the other is R-type. S-type bacteria is that kind of bacteria which have polysaccharide capsule. Its colony appears as smooth and shiny, so he named it S-type. And the other type forms a rough colony due to the absence of polysaccharide capsule in it. So he called it R-type bacteria. Let's see what kind of experiments he performed during his research. In this diagram, I will show you that what kinds of experiments were performed by Frederick Griffith. First, he injected R-type strain into a group of mice, and as a result, all the mice remained perfectly healthy. So he made conclusion that R-type strains are non-virulent as they are not causing any disease in mice. And then he injected S-type strain bacteria into another group of mice, and as a result all the mice died. So he made conclusion that S-type strain is virulent. Then he injected heat-killed smooth strain S-type bacteria into another group of mice, and as a result all the mice remained perfectly healthy because the S-type bacteria are killed, so they did not cause any disease in mice. Next, he performed another control experiment in which he mixed R-type bacteria with the heat-killed S-type bacteria. R-type bacteria and heat-killed S-type both are non-virulent, so he was expecting that mice will remain healthy. But the result was quite opposite. When he injected the mixture of R-type strain and heat-killed S-type strain into a group of mice, the mice died. They got the disease and undergoes death. So he found and he made the conclusion that this is because of transformation. Transformation is the transfer of genetic material from one organism into another by which genetic makeup of recipient is altered. And Frederick Griffith's experiment concluded that the information in the polysaccharide capsule and virulence had passed from the heat-killed S-type bacteria into R-type and making or converting the R-type into S-type because it is becoming virulent. This is known as transformation. But here, the agent which is responsible for transforming R-type into S-type still undiscovered until 1944. Avery, macleod and mccarty were the scientists who found out the agent responsible for transforming R-type bacteria into S-type bacteria. They were the one who found that DNA is the hereditary material. They made an extract filtrate of heat-killed S-type bacteria mixed with R-type bacteria. And their extract filtrate have carbohydrate, lipids, proteins and DNA. Then they divided this extract into 4 samples. These 4 samples. In first sample, they injected the extract filtrate into a group of mice, and as a result all the mice died. It means that transformation had occurred and the filtrate of heat-killed S-type bacteria have the active factor which is responsible for the transformation. Next, the second sample of the extract. They treated this sample with the proteases and removed all the proteins from it. And then they injected this filtrate into a group of mice, and as a result, all the mice died again. So they found out that protein is not responsible for transformation, because in the absence of proteins, transformation still occurred. Then, third sample of the extract was treated with RNases. They removed the RNA from the extract filtrate and then again injected into another group of mice, and as a result, all the mice died. So they found out that RNA is not responsible for transformation, because in the absence of RNA, transformation still occurred. Then they treated fourth extract sample with DNases and removed all the DNA from it and then injected into another group of mice, and as a result, mice remained perfectly healthy. So they made the conclusion that transformation had not occurred when they treated the extract filtrate with the DNases. It means the DNA is responsible for the transformation. DNA is the hereditary material. So this is how it has been confirmed that the active factor is DNA, which is causing the transformation of live R-type into S-type bacteria. I hope you like this video. Please like and subscribe this channel.